Okay, so welcome to another session of bioinformatics. I am Dr. Y.V. Rajesh, Department of Biotechnology. In this session, we will learn about origins of phylogenetics. So, phylogenetics basically two types: classical phylogenetics, and another one is molecular phylogenetics. Here, classical phylogenetics and molecular phylogenetics here. So molecular phylogenetics, it came because we learned that DNA is the genetic material and proteins are derived from the DNA sequence information here. Molecular phylogenetics is a branch of phylogeny that analyzes hereditary and molecular differences using DNA sequences to gain information on organisms' evolutionary relationships. Whereas classical genetics, classical phylogenetics, we use the physical characters here, physical aspects. Suppose if you take the plant here, the type of the flower and the size of the flower and the arrangement of petals, arrangement of sepals. So these physical characteristics are observed and trees are constructed here. Whereas here we use simple DNA sequence here. So it is considered as a one of the biggest development in biology to understand the evolutionary process here. So we know that according to Darwin and according to modern genetics, the living organisms or the evolution happens because of 5 different processes here. So the process of 5 processes are: mutations will happen in DNA. So DNA undergo mutations. A will change to C, C will change to G, G will change to T, vice versa here. Mutations, because of mutations in functional regions, lead to new characteristics. The new characteristics will use to new organism or species here. And genetic recombination. So during the mitosis and meiosis process, at the time of cell division, the genetic material will exchange among the homologous chromosomes here. So sometimes the problem with the genetic recombinations lead to chromosomal aberrations, then that lead to the evolution of new species or unwanted characteristics here. Similarly, the chromosomal organization. So the chromosome generally organized into euchromatin and heterochromatin, and the organization also depends upon the external features here, external cues. The external cues might disturb the chromosomal organization, and also epigenetic natures. Epigenetics also play chromosomal organizations here. So these three are at the level of DNA and chromosomes. And you have another gross process, natural selection and reproductive isolations. All these 5 processes leads to the evolution of living organisms here. Basically the molecular phylogenetics or bioinformatics, it focuses on mutations and DNA and protein sequences. DNA and protein sequences here. So the molecular phylogenetics basically advanced because of Frederick Sanger. In 1953, Frederick Sanger and his colleagues determined the primary sequence of insulin protein. All the amino acid sequences of insulin protein has been sequenced using protein sequencing technology, and he is the first person who explained protein sequencing and also DNA sequencing, and he got 2 Nobel Prizes here for his great accomplishments in molecular biology here. So using the sequences of insulin here for different organisms: human, chimpanzee, rabbit, dog, horse, mouse, pig, chicken, sheep, bovine, whale and elephant. So by performing multiple sequence alignments, we can derive some hypothesis called as molecular clock hypothesis. So molecular clock hypothesis, simply based upon the number of mutations for 100 amino acid residues in homologous proteins or closely related proteins here. So the idea is very simple here. So the rate of nucleotide substitution or the rate of mutation. So the rate of mutation can be given as number of mutations per 100 characters divided by 2 into time in million years here. So the rate of substitution and number of mutations. So from this one you can calculate the years of evolution here. So basically we will get K from simple comparisons here. So R can be constant for different proteins here. So from this one we can get the values of T here. So for a given protein, for a conserved protein, so the rate of substitution is constant. So the rate of what do you say? So the evolutionary rates also will be constant here. Evolution timelines also will be constant here. So some of the proteins will undergo mutations very quickly, and some of the proteins does not undergo mutations here. Suppose fibrinopeptides which are present in the blood here, okay, and milk proteins and albumin proteins of bloods, and these proteins actually they undergo mutations. So that means they change in different organisms here. Whereas histone H4, the ubiquitin protein, histone H2B, so histones are involved in nucleosome organizations, and cytochrome in energy metabolism. Insulin is a hormone which is in glycolysis and glycolysis pathways here. So these are most conserved. So the number of mutations are less for these sequences here. So by using simple comparisons we can shed light on the evolutionary paths of the living organisms here. So this forms the basis for molecular phylogenetics, and it is known that the coding sequences should not be mutated. So their rate of mutation is 1.58 per billion year, whereas non-coding sequences such as introns or lot of junk DNA present in genomes here, and promoter regions and other regions, the rate of mutations are quicker compared to coding regions here. So with these basic ideas, so the molecular phylogenetics is carried out here. So thank you for listening to this session. In today's session we learnt about why molecular phylogenetics is studied in bioinformatics. The reason is, simple reason is, so every living organism came from a common ancestor, and they have DNA as a genetic material, and there are traces of evolution in the DNA sequence. Thank you very much.